The goal of this assay is to chemically denature HCA2 in such a way that we can measure thermodynamic parameters in order to determine whether your mutation caused a decrease in the protein stability. So, how will we do this? The assay utilizes a 96-well plate so that we can test many conditions in a compact and efficient format. The top 2 rows of the plate will be for blanks, the next 3 for wild type, and the bottom 3 for mutant. Using 3 rows allows us to test each condition in triplicate for the wild type and mutant protein. In addition to protein or buffer, urea will be added to each well. The easiest way to organize the plate is to have each column be a single urea concentration. Again, this allows the experimental samples, the wild type and mutant protein, to be measured in triplicate. We can add 0 molar urea to one end of the plate, and then increase concentrations as we move across the plate. Let's take a look at what's happening in a well with wild type protein and no urea. Here you can see HCA2 fully folded and floating happily in solution. The way that we will measure just how happily folded each protein sample is using fluorescence spectroscopy. You can see here that the tryptophan residues in the HCA2 protein have been shown as spheres. Tryptophans can fluoresce by absorbing light at 280 nanometers and then emitting light at around 325 nanometers. We can use a fluorimeter to measure that fluorescence. So now let's see what happens when we move to the middle of the plate where the urea concentration is considerably higher. You can see that the HCA2 is partially unfolded and those tryptophan residues are more solvent exposed. When these tryptophans are excited at 280 nanometers, their emission is actually shifted so that the peak of that emission is closer to about 335 nanometers. This shift in the peak fluorescence wavelength allows us to follow along with the unfolding from urea concentration to urea concentration. If we jump to the very highest urea concentration in the rightmost column, now we can see that HCA2 is completely unfolded and all the tryptophans are fully solvent exposed. When these tryptophans are excited at 280 nanometers, their peak emission is actually around 345 nanometers, so shifted even farther. As we've seen then, what we are measuring is the peak emission of the tryptophan residues and how that peak shifts as the protein is gradually unfolded with gradually increasing concentrations of urea. We are able to do some normalization of this fluorescence data to convert it into fraction folded, which tells us on a scale from 1 to 0 how well folded the protein is at each urea concentration. When we plot those numbers, we get a sigmoidal curve, like this. This curve is useful in determining the midpoint of unfolding, or C sub m, which is the urea concentration where the fraction folded is 0.5, or half. This data can also be processed further using the equation delta G of unfolding is equal to negative RT times the natural log of KEQ to determine the free energy of unfolding of your protein. So what you do when analyzing this data is extrapolate the linear region of this fit to 0 molar urea. And this value tells you the energy required to denature your protein when it is in buffer. Because this is the delta G of unfolding, a positive value means that the unfolded state is less favored than the folded state. So a larger, more positive delta G of unfolding indicates higher protein stability. It can also be useful to recognize that the C sub m value is the urea concentration where the delta G of unfolding is equal to 0. Let's go back through the experiment a second time and consider a mutation that causes a significant decrease in protein stability. Remember when we looked at wild-type protein in 0 molar urea and it was happily folded. Now let's look at the mutant as well, which will be shown in magenta. It is also happy at 0 molar urea, so just like wild-type, when these proteins are excited at 280 nanometers, their peak emission is around 325 nanometers. When we move to a moderate concentration of urea, remember the wild-type HCA2 was partially unfolded. This mutated version of the protein, by contrast, is significantly more unfolded. That means that the mutant's fluorescence emission will be more shifted than the wild-type's. That increased shift corresponds to a lower fraction folded, so that when the data is plotted for the mutant, the curve might be shifted slightly as compared to the wild-type, like so. And when this data is converted to delta G, you can see that the y-intercept is lower, indicating a less stable protein. So you will be using this experimental setup and going through a number of data analysis steps to ultimately be able to determine whether your mutation has affected overall stability of HCA2.